Hey everybody, my name is Jason Walker and I am a biology coordinator with the National Math and Science Initiative, and this is the third screencast in the biotechnology unit, and this one is going to investigate genetic engineering techniques. And here's just a little funny thing that I saw. Genetic engineering, sometimes we can go too far. Now this is just, this is a joke. This is just a fake picture, just of this chipmuncula, right? Half chipmunk, half tarantula. But I want you to. I like what it says is sometimes you go too far, right? And as we go through here, I just want you to be thinking. We don't live in a society anymore where the question is, can we or will we? It's should we, right? And you get to make that. You get to make that decision. So as we go through and look at all of these genetic engineering techniques, I want you to ask yourself, is this okay with me? Does this cross my line? Okay? And it's okay if some of these cross your line. It's okay if somebody else, if their line does not get crossed by a particular process that maybe you have an issue with. But that's the society that we live in, right? We investigate things in biotechnology or genetic engineering, things that we're able to do today that we weren't able to do even 10, 15 years ago, right? So let's look at biotechnology. It's the manipulation of natural biological processes in order to serve societal needs. Okay? So we just look at basically what's going on. We have a bacterial chromosome that has a particular plasmid. Okay? We have a gene of interest. Let's say that this is one of our cells. Okay, you can see this is a eukaryotic cell. This is a prokaryotic cell. So we cut the gene out. We incorporate that into the bacterial plasmid, this little circular stretch of DNA right there. Okay? Through transformation, it's going to take that back up, right? It's going to start replicating, and then we can use that human gene. We can use that to start making things like human growth hormone, right? Protein that dissolves blood clots and heart attack therapy, right? It doesn't have to be a human gene. It can be genes that are used to alter bacteria for cleaning up toxic waste. A gene for pest resistance inserted into plants, right? And so we're able to facilitate processes. Natural biological processes. We are manipulating them to serve a purpose for us. And you may or may not think that that's okay. And I encourage you to get all the facts and just think about, does this cross my line? Let's think about the four different types of biotechnology. Okay, four main areas. First one is transgenic biotechnology. It's the mixing of genetic material from multiple sources. So if you have a bacterium that now has a human gene in it, okay, transgenic biotechnology right there. Also reproductive cloning. We are cloning certain species. Okay? Now we've been cloning things like plants, things like that for years, but now we're starting to do things like we're going to clone sheep, clone cats. We're cloning some mammals here. Okay? Reprogramming of cells, third type. Reprogramming of differentiated cells or using stem cells to become needed tissues in patients with diseases or physical harm. Okay? And the last one, of course, is what has been made popular by shows like CSI, NCIS, that type of thing. Forensic biotechnology. The use of restriction enzymes and electrophoresis, and you probably have done this in class, and to distinguish one person from another. Okay? So, again, where is your line? As we go through here, I just want you to find your line. Okay? I'm going to present you with a lot of different techniques and examples of how biotechnology is being used in our society. Okay? It's your job to decide how far is too far. Where is your ethical line and what factors dictate when your line is crossed? Okay? Remember, if your line is different from somebody else's, it's perfectly fine. So let's look at transgenic biotechnology. Okay? So the pituitary gland produces a crucial hormone called the human growth hormone. Okay? Now, if you are of normal height, your pituitary gland is making HGH just fine and everything's perfect. Not everybody is born that way. Okay? So what they used to have to do in order to help these people is that they had to extract it from the pituitary glands of deceased humans. Well, again, you had to wait for somebody to pass away in order for that to occur. So we didn't have enough. Okay? Very expensive. HGH is a peptide hormone. Now, peptide makes you think it's protein. It is. And it provides for normal growth and development. Okay? If the pituitary gland is defective, then growth is stunted. Okay? Starting in the mid-1980s, okay, we started to facilitate a process where we can get all the HGH that we want. Okay? So what we're doing is we're making recombinant DNA. Okay? The HGH gene is cut out of a human genome and it's inserted into the plasmid, like we saw a couple of slides ago. Now that's recombinant, what we call recombinant DNA, because it has DNA from multiple sources, one from the bacterium and from humans. Okay? The plasmids then take those up via transformation. It's just the uptake of DNA outside of yourself into your cell. Okay? And then as the bacterium starts to reproduce, the bacterium undergoes transcription and translation as well. It makes all the HGH that we could possibly need. Okay? So as you look here, I want you to think. Recombinant DNA is made here. Okay? How is it made? How is recombinant DNA made? Well, we took a human gene, cut that out with some restriction enzymes. We incorporated that into, we took the same restriction enzyme, cut a hole in that plasmid, then stuck that human gene in there. Okay? We inserted that gene into the plasmid. Transformation incorporates that into the host cell. Okay? That's recombinant DNA. That's how that works, right? So where did the plasmid come from? The plasmid came from where? The bacteria or the prokaryote, right? What is transformation and what is the role it's playing in this process? Okay, and we just talked about that. Transformation is just the uptake of that DNA outside of yourself into your cell, right? You have to be able to take that plasmid and get it back into the bacterium or another bacterium in order for it to start facilitating processes like transcription, translation, so it can make this protein, this HGH. Okay? Now, let me just stop here. Does this cross your line? Are you okay with us using bacteria, cutting genes out of our genome, incorporating that into a plasmid from a bacterium, and having that bacterium make human protein for us? Is that okay with you? Okay? So just think about that. So again, a plasmid is just a small circular piece of DNA that not only is separate from the chromosome, but it can replicate independently. Okay? So how do they cut the gene of interest out of the genome? Okay? A portion of the DNA strand, which is highlighted in white here, okay, has recognition sites. And here we see G, G, A, T, C, C. Okay? So a restriction enzyme comes and finds that G, G, A, T, C, C, and it makes a cut. Okay? It makes a cut here, and it makes a cut here. Okay? With that second restriction enzyme. Okay? And that makes what we call sticky ends. Okay? Have a little bit of where you can place this back on if you want to. Okay? And it'll fit just perfectly. Okay? So basically, the restriction enzyme cuts that sugar phosphate backbone. Okay? DNA fragment from another molecule cut by the same enzyme. You see that it fits perfectly in there with these sticky ends. No sticky ends, this new fragment won't stick in. So if this is the genome of, let's say, the plasmid, okay, if this DNA right here is from the plasmid, we cut that and we stick in the gene of interest in right there, okay? And then DNA ligase is that enzyme, okay, that's basically just going to glue those back up. So now you have a recombinant DNA strand with DNA that comes from multiple different spots, okay, multiple different organisms. So let's talk about insulin for a second, okay? Another organ, the pancreas, among other things, produces insulin, okay? So what does insulin do? Okay, it ensures it's another protein hormone. It's another peptide hormone. It ensures that glucose is taken up by cells for cellular respiration. Okay, and those individuals that have diabetes, okay, let's say that pancreas is defective, then the blood sugar levels get dangerously high in the bloodstream. That's why we look for that's why they have to constantly test their blood sugar, because without insulin, that glucose, okay, it's unable to go from the bloodstream into the cell. Now, once it gets in the cell, it's used in cellular respiration to go through glycolysis, Krebs cycle, electron transport chain. You're making all this ATP undergo, i.e., cellular respiration. Okay, so they need the insulin. Okay. So, using very similar techniques as what we just looked at with the human growth hormone production, scientists were able to use E. coli to bioengineer synthetic insulin. Okay, in 1977. So that's actually not that long ago, right? Some other transgenic organisms are used to produce insulin today, such as yeast. And also the safflower plant. So this plant that you see here is actually we are actually able to get bioengineer synthetic insulin that for diabetics, okay, using these plants. So I ask you, does this cross your line? Is it okay for us to bioengineer, use yeast, use these plants, use E. coli, or is that okay? Does that cross your line for somebody with diabetes to have all the insulin that they need to be able to survive? Right? Now again, I just want to stop here and just say, if that crosses your line, that's fine. Okay? But again, we're just trying to find where is our line. Okay? So let's talk about golden rice. Okay? The World Health Organization estimates that somewhere between 1 to 2 million children die every single year from vitamin A deficiency. Okay, vitamin A deficiency is usually if you live in the United States, there's a tremendous amount of vitamin A. If you have anything with vitamin A or even beta carotene in foods will give us the vitamin A that we need. Okay, to help us to not lose our eyesight. So what somebody did is they bioengineered a particular type of plant, particular type of rice called golden rice. And it's been genetically modified with beta carotene. So it has the vitamin A that these kids, predominantly in third world countries where we are sending them rice, we're sending them food anyway. Okay? And it has the beta carotene that they need, and these kids aren't going blind anymore. Okay? Now this transgenic organism is the result of mixing genes from a bacterium and from daffodils. And you are mixing those genes into the rice genome. Now, because of that, when you eat that rice, you're now getting the vitamin A that you need. Okay? Now, I want to say, it's currently not being used. It's been outlawed because of regulatory issues. Okay, so now we start thinking about things like what you may see in the news: Frankenfoods, GMOs, these genetically modified organisms. There's a huge debate right now going on about these genetically modified organisms. Is it okay to eat? Is it not okay to eat? Am I okay with you eating it, but maybe I don't want to eat it? Right? So what do you think? Do you think that we ought to be able to use this? What is your educated opinion on this? I want to keep stressing. If you don't think that we ought to be able to send golden rice to third world countries for these kids, that's fine. If you do, that's also fine. Again, we're just finding our line. Okay? Cool. So let's talk about reproductive cloning. This is one of my favorites. So what is a clone? It's an exact genetic replica of another cell or another organism, right? So what have we cloned so far, right? We can clone DNA through the polymerase chain reaction that we'll talk about here in a little bit. We have cloned cells by creating tissue cultures or stem cell lines. And we've also cloned whole organisms, okay? So let's talk about these whole organisms. What have we cloned thus far? Well, we've been cloning plants for thousands of years: bananas, potatoes, grapevines, through grafting, that type of thing. I come from a very rural community in Arkansas. Okay, you can probably tell from my accent that I'm I'm Southern. Okay, so but basically we have a lot of vineyards where I'm from. Okay, and we were able to take the grapevine, the grapes that we wanted, okay, and but there was a bunch of microorganisms and some disease that was caused basically in the roots, and so we couldn't grow the grapes, the grapevines for the wine that was being made effectively. So what we were able to do is we were able to graft a particular rootstock onto the grapevine itself, right? And that root was immune to that disease. So now we had rootstock that was immune to the disease, and we're still growing the grapes that we want. Okay? So it's perfect. It's a perfect marriage there. So we've been cloning trees, shrubs, vines, that just type of thing. Okay? So we've been doing that for hundreds of thousands of years. Okay? So let's look at animals. Parthenogenesis. Parthenogenesis is literally the asexual reproduction that occurs naturally where offspring is born without sexual reproduction. This is supposed to say born without. Okay? So it's born without sexual reproduction. Okay? So basically we've seen this in some insects, anteaters, sharks, and a lot of different organisms. Okay? So basically you don't need the male. Much like all cloning, you do not need the male. Okay, so let's look at sharks just for a second. Parthenogenesis is basically the mother, through oogenesis, makes the egg, right? The meiosis. So she's making the female gametes. You know how you have the egg, and then you have those three polar bodies. Well, one way that you can make a zygote is you take one of those polar bodies, and you just fuse that with the egg. Well, now you have a diploid organism. That's parthenogenesis. This naturally occurs. This parthenogenesis is not something that we did. Okay? This naturally occurs. This is naturally occurring cloning. Okay? Then also some animals can undergo somatic cell nuclear transfer. They've undergone this somatic cell nuclear transfer, such as sheep, rats, cats, goats, dogs, camels, many other things. We'll talk about that. Okay? So let's talk about the somatic cell nuclear transfer. Okay? So this is how we made Dolly. Dolly the sheep. Okay? There she is. So basically what we did, and by we I mean humans, we took a cell from her mammary glands, okay? And we took the nucleus. We took the nucleus out of just a regular somatic cell, a diploid cell. We took the nucleus out, okay? We took the nucleus from one of those mammary cells, okay? And we injected that, okay? So now I have a nucleus from a mammary cell, okay? I grew that in culture, found a surrogate, and that embryo grows. Okay, you implant that in the uterus of the surrogate sheep, the third sheep, the surrogate mother, and she delivers the clone. Now you think we never ever at any point mixed genes. It's the exact same chromosomes. The exact same chromosomes that was in one of those mammary cells. Right? So if we did that, if we're talking about human cloning, so we just find a regular, just somatic cell, we take the nucleus out, 46 chromosomes, right? So we implant that into that empty cell, and then we just let that mitotically divide, implant that in the uterus of a surrogate mother, and then that is the clone of where you got the cell from. Now I just want to say this. We always think about, oh, well, I'll just clone myself and there'll be two me's, right? Well, if you're 16 years old and you clone yourself, that clone has to be born. So it's always going to be 16, 17 years behind you, right? Okay. So what do you think about this? Do you think it's okay for us to clone sheep? Well, let me give you another perspective, okay? What if we were to manipulate animal embryos and use recombinant technology to give these animals some beneficial characteristics? Beneficial to who? Well, us, right? We humans. Well, this is what some scientists have been able to do. Some animals, like this goat, have been bred to produce certain peptide hormones, like human growth hormone, remember? And they express those in the milk. These proteins can then be easily separated from the milk for human use. So now you don't have to worry about the bacteria. You can genetically engineer sheep, these goats, to just naturally express whatever protein, whatever human protein you want them to. So what about this? This is a much cheaper way to get human growth hormone and other things that we desperately need. Well, some people desperately need to be able to survive. So much cheaper way to go about this. Just extract it from milk, right? So what do you think about this? If you knew somebody that had these debilitating diseases and they needed these medicines, are you in favor of this type of technology? And again, if it's yes, if it's no, that's fine. But let's find our line. Where is our line? Okay? Cool. So let's talk about reprogramming cells. Okay? Huge debate now. Stem cells and stem cell research. Okay? So what can stem cell research do for me? Do for us, right? So organ and tissue regeneration, fighting off diseases like cardiovascular disease, Parkinson's, Alzheimer's, blood diseases like leukemia, sickle cell anemia, right? And you think, well, if stem cells can do all of that, so what's all the hubbub about? What's the big fuss about? Okay? The stem cells that work the best come from embryos, right? So getting them from embryos, that's that. that's where it becomes a sticky situation. That's where the debate comes. We don't want to use embryos. Those embryos theoretically could become a human, right? So let's just talk about totipotency. Okay? Plant cells are totipotent, which means that their cells, if you were to, any cell can become any other type of cell. Okay? So, for instance, if we take a carrot, fragments were cultured in nutrient medium. Okay? We chopped them all up, right? Got single cells out of there. And then we basically took care of those cells, right? They were actually those single cells were actually able to divide. Okay, the embryonic plant developed from a cultured single cell. The whole plant had the ability to make a whole organism. A single somatic carrot cell developed into a mature carrot plant. Everything that plant needed to be an adult plant was in that one single cell. That's totipotent. Okay? So let's talk about stem cells. They are undifferentiated, meaning that they haven't become a type of cell yet. Okay? I always just very loosely describe it as they don't know what they want to be when they grow up yet. Right? So when a sperm meets an egg, the resulting zygote is totipotent. The inner cell mass, the source of embryonic stem cells, are pluripotent. Okay? So let's investigate what are these two things. Totipotent cells have the ability to create a whole organism, or at least all different types of tissues. Pluripotent cells can only give rise to most types of tissues, but definitely not a whole organism. Okay? So let's talk about these iPS cells. Okay? In 2007, the induced pluripotent stem cell line were developed. Basically, what we were able to do, okay, is reprogram genes. Okay? We were able to reprogram and make our own stem cells. Reprogrammed genes are spliced into a normal human somatic cell. Okay? This tricks the cell into changing from a differentiated cell into a pluripotent cell. The cell can then develop into a desired differentiated cell of another type. Okay? So let's say that you want a stem cell to become, let's say, a nerve cell. Okay? So we have reprogrammed genes that are spliced into this virus. The virus is then allowed to inject these into, let's say, human skin cells. Okay? Viral infection of skin cells causes the insertion of reprogramming genes into the skin cell DNA. Okay? Added genes reprogram the cells, turning them into iPS cells, these induced pluripotent stem cells. Factors, so now it's a stem cell, so factors then added to that allow them to produce cells of a particular desired type. So now we can actually turn a stem cell into a nerve cell. We can actually take a differentiated cell, reprogram it, and make it into another type of cell. Why is this so huge? Do you remember the whole debate on we don't want to use embryos to get our stem cells? This will eliminate that if we're able to use these. Okay? This eliminates the need for embryonic stem cells. In 2012, the Nobel Peace Prize in Physiology and Medicine went to these two gentlemen right here. They were able to, in their labs, okay, through both of their work, okay, they were able, both their work developed, led to the development of iPS cells, okay? So let's look at forensic biotechnology, okay? Forensic biotechnology is used to determine the identity of certain individuals. Okay, criminals, disaster victims, biological parents, that type of thing. Okay, and what we use here is gel electrophoresis. Okay, so basically you have a mixture of DNA molecules of different sizes. And you've probably done this in your class with your teacher. Okay, so I just want to engage you kind of in a discussion of what this process is. Okay, so let's just think about what you've done in your classroom. So you put the DNA in these wells, right? You hook that up to a power source, negative cathode, positive anode over here, right? And you turn that on, and the longer molecules will stay down here, while the shorter molecules will migrate all the way down here. So basically, take restriction enzymes, you're cutting up that DNA into different segments. The longer fragments are going to stay up here. Shorter molecules, the shorter fragments of DNA are going to migrate down here, and you can be able to, and you will be able to compare one sample of DNA to another sample of DNA. This sample of DNA was found at the crime scene. Here's the sample. Here's suspect 1. Do those DNA molecules match up? Right? So let's think about this. Why is the DNA being placed on the negative side? Well, if you look at a DNA molecule, right? DNA with those phosphate groups, remember DNA is made up of nucleotides. It's a nucleic acid. It's made up of nucleotides. So each nucleotide is made up of 5-carbon sugar, that pentose sugar, nitrogenous base, and a phosphate group. And a phosphate group is negative, has a negative charge. Therefore, DNA is negative. So if I put it on the negative side, it will migrate down to the positive side. Does that make sense? Okay. So this gel that we use is kind of like a matrix of tunnels, okay? And so the smaller ones are going to be able to migrate farther, and the larger ones are going to kind of get stuck up here. That's why the larger molecules are up here, shorter molecules are down here. Okay? So one way to analyze a sample of DNA via electrophoresis involves cutting the sample of restriction enzymes like we've said. This requires a large sample of DNA, such as from a test tube of blood. Cutting with the enzymes that will produce fragments of DNA. Okay? The length of those fragments will differ between individuals. Those differences are called restriction fragment length polymorphisms. Okay? RFLPs. Okay? So this illustration, this slide, and the next could be RFLPs. Okay? So let's talk about the polymerase chain reaction. Okay? Usually if there's only a small amount of DNA to work with at a crime scene, so what we need to be able to do is just replicate that. So investigators and forensic scientists use PCR, polymerase chain reaction, to make thousands and thousands of copies of key regions of the original DNA strand. Okay? So let's look at PCR. Okay, so basically you have a DNA strand that you want to replicate. So you're going to heat that briefly to separate the DNA strands. All right, then you're going to cool it and allow primers to hydrogen bond where they need to go. Okay, and then you've also added in some DNA polymerase and some nucleotides that adds nucleotides to the 3' end of each primer. Okay, and then replicates that all the way down. Okay, continues DNA synthesis all the way down. You keep doing this, and you keep making. See, we went from 1 to 2 to 4 to 8, right? And you keep doing this process, and it makes more and more and more DNA. Okay? So it's important to realize that only a relatively short, probably up to about 20,000 base pairs, but typically shorter than that, are the target sequence. Typically, it's relatively short. It's actually short amount. It's actually being amplified. The area between the two primers is the target sequence. The chromosome goes on for a very long distance, both above and below the stretch being shown in this illustration. Okay, so it's not just a short amount of DNA. Okay. PCR can be used Let me say this. It cannot be used to copy an entire chromosome. Okay? So as you look at this, just kind of stop here if you want to. Just kind of pause it. And I want you to answer the following questions. Okay? Kind of research this if you don't have this. But what's the primer used for? How is DNA polymerase used in this process? Number 3, what else must be in the test tube for the process to work? Okay? What does heating up the test tube mixture do? And what does cooling the test tube mixture do? Okay? So you might just make sure that you can answer those 5 questions. Okay? And just think about this process. Now, you will not have to explain this process on the AP Biology exam. Okay? But a good understanding of this will allow you to use it as an example and talk about DNA replication, DNA synthesis, DNA polymerase, and primers, and all of this, and will allow you to use this as an example of how we humans are able to manipulate DNA to serve a purpose for us. Okay? So, let's stop here just for a second. So we've looked at all types of cloning, all types of transgenic bacteria, all types of turning differentiated cells into undifferentiated cells, i.e. Stem cells. Where are you at? Are you okay with all of this? If not, which ones have crossed your line? And think about why. What is it about that crosses your line? Is there anything that you would change about any of these processes that would allow it to not cross your line? Okay? And again, there's no right or wrong answer to does it cross your line, does it not cross your line? Okay? Just what I'm trying to do is just get you to think about where is your line? Where do you follow? Where do you fall on that? Okay? So let's think about electrophoresis here, okay? Each PCR reaction typically amplifies at a single loci, okay? A single locus. Since we all have 2 copies of each locus, thanks to our maternal and paternal homologous chromosomes, we will get 2 separate products from a single PCR reaction. If the region between the primers on our paternal chromosome differs in length from the region between those same primers on our maternal chromosome, then we get products of different sizes, which then will show up as 2 separate bands on a gel. Okay? If the products happen to be the same length, then only one band results, because it'll be at the same place, right? One of the most common regions for PCR analysis are those containing short, repeated segments of DNA between the primer sites. These are called STRs. Okay, short tandem repeats. One might have 15 repeats of that sequence between the primer sites on the paternal chromosome and 18 repeats of the same sequence between the same primer sites on the maternal chromosome. This would then result in 2 bands on a gel when these PCR products are analyzed. By using multiple sets of primers, one can get numerous bands to compare between samples. Numerous other differences could be analyzed other than STRs, such as SNPs, or single nucleotide polymorphisms. But students do not need to know every possible method of analyzing DNA differences. They do need to know that the differences exist and why they exist. Okay, so you as a student don't necessarily have to know every possible method of analyzing this. Okay, but knowing the differences is going to be integral. Okay. Now, this is Earl Washington. Earl Washington was sentenced in 1982 for rape and murder. In 1994, DNA from the crime scene was tested, and in the analysis of the DNA on the victim and the 2 suspects, it was found that Mr. Washington was not guilty. Shortly before his execution, the governor of Virginia gave him clemency and commuted his sentence to life in prison. Years later, he was given a full pardon and released from prison. Now That's after 17 years. So in this table, the numbers refer to the number of repeats at each locus. For instance, at locus 1, SDR marker 1, Earl had 16 repeats on one of his chromosomes and 18 on the other. The evidence DNA had 17 and 19 repeats, meaning it could not have come from Earl. But it did match Kenneth Tinsley, another possible suspect. Note that SDR marker 1 was enough to eliminate Earl as a suspect, but that single site would not be conclusive as it is coming from Kenneth. Numerous sites were used and all matched Kenneth Tinsley, and he was convicted. Now, so here's possible DNA analysis here. Okay, so we have the ability, to sequence DNA. We have the ability to get a particular gene and get a particular stretch of DNA, and we can sequence that and figure out in what order are the A's, G's, T's, and C's. Right? So we have the ability to sequence that DNA. This means that if we know the gene that we're looking for, and we can analyze someone's DNA for a specific sequence or allele, therefore we can tell you definitively, let's say for instance, if you have a disorder like Huntington's, which is an autosomal dominant disorder or not. We know what the Huntington's gene is. We know that it's caused by repeats of CAG, 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 CAG. Now, if you have under a certain amount of CAG repeats in a row, that's the recessive allele. But if you have over a certain amount, now that gene is the dominant allele, and now you will have Huntington's. And so, Huntington's is a degenerative brain disorder that usually starts causing telltale symptoms around 35. There is no cure, and it's eventually fatal. So the question is, if we can analyze your DNA, would you want to know whether you have it or not? Right? Okay. So think about this. If you wouldn't want to know, let me put another little wrinkle into the story here. If you wouldn't want to know, what about your children? Let's say you're heterozygous for this disorder, right? Well, if you're heterozygous, every single time that you have a child, there's a 50% chance of passing that on to your child, right? This is a autosomal dominant disorder, which means that if you have the big H, you have it. And it doesn't matter whether you're autosomal dominant or heterozygous, you have it. So now, does that change your mind? Now would you want to know? Because maybe that affects your decision to have kids or not. Again, whether you choose to have kids or not have kids isn't the point. The point is that we now live in a society where we can tell you things. We can tell you something about your DNA, so now you can make more informed decisions. Right? So would you want to know? So this whole time we've been talking about different processes. And I've been asking you, would you want to know? Okay? Let's just look at some other real-life examples of biotechnology that we haven't talked about yet. And just think, am I okay with this? Okay? Injecting human brain cells into monkey brains. This is for brain disease research. Does that cross your line? Now what about xenotransplantation? This is when we use animal parts for our parts. For instance, using a pig valve to replace a defective heart valve in a human. Now, I'm very sad to say that the pig valves that work well aren't the adult pigs, the little cute ones. Okay? So let's talk about Bt crops. Okay? These crops contain genes from a particular bacterium which produces proteins that are toxic to insects. So are you okay with your crops, these crops that you're eventually eating? Are you okay with those genes being in there? These genes from these bacteria. And what about Roundup Ready crops? These crops contain genes that protect them from Roundup. And so if a farmer is going to be able to just kill off all the weeds, right, with this herbicide, but it doesn't affect the plants that the farmer is wanting to grow in order to sell to you to eat, right? Are you okay with these things? So again, the point of this screencast was just to show you some different techniques, things that we're able to do now that we've never been able to do before, and just get you to start thinking about, we don't live in a society anymore where the question is, can we? Or even will we? We can. And we will. But the question is, should we? Right? And that's a decision that we all make. We all get to make as informed citizens. And so what I encourage you to do is find out all the facts that you can, and then make an informed decision, whatever that decision might be. Okay? Now, this is the final screencast for biotechnology. Okay? I hope that you have learned a lot, enjoyed a lot, and if you have any questions about any of this, I want you to make sure that you contact your teacher and ask any questions that you can. Okay?